So good? Okay. So first one is preclinical. Walk into a room. We work with a lot of small companies, so sometimes they're actually academics or faculty members or people who are spinning out of universities. So they're more on the research end of things. And our drug is extremely safe. We tested it on mice in our pharmacology study for 90 days, and those mice were still happy at the end. Good. Or else here, like, oh, and our drug is really safe because the study was completely clean. I hear this all the time of people sort of espousing that their drug is completely safe based on sort of limited data sets. I'll see it in grants, I'll see it other places. And this is one of the biggest myths because, Laura, I know we've talked about this. 3 months later, after we're espousing the safety, we're like, have dead dogs. I mean, that's terrible to say, but we don't say that until we've actually done the studies and proven our safety. And so the whole point here is you can't actually make the claim that your drug is safe until you've actually established what is going to be your safety margin.
Or until you've actually seen toxicity.
Yes, or you've identified the toxicity. So I'm as nice as I can be to the folks and say, well, it might seem safe, but here's the questions to ask. Have you looked at the animals' histopathology? Have you actually, they were in a pharmacology study, did you actually open them up and look at each tissue and make sure that the tissues look good? So that's the path part of it. Not everybody does. Some people do legs up, legs down. That's a very gross analysis of health. Have you defined the dose-limiting toxicity? So you said your animals were happy and healthy, but did you actually find anything that's going to help you establish that? This is the big one. Coming off of a pharmacology study, did you look at suprapharmacological doses? Because you were working at efficacious doses in that study, so you were getting the health of your animal at efficacious doses. But what we want to know is when you overdose that animal, what happens, right? That's the point there. So driving all this is sort of our transition of when we start working with our groups back at that sort of late discovery phase. They're making these claims off of this sort of limited data, right? You might have an effect, but have you determined that your drug is druggable? You might have an effect, but do you have a marketable or competitive product? You might have an effect, which is fantastic, but have you actually defined the acceptable therapeutic index to move this product forward in the range? So this is these guys. I'm talking about these folks. Now, I apologize if some of you are not in this range. Some of you might be further along, but this point is coming in with my sort of folks that come in this late discovery phase where they found a scaffold that's active. They found a target that's working, and they're seeing some either binding data or early pharmacology data. At this point, this is where I get up on my soapbox and say, we need to develop a target product profile. That's where we need to start here, develop what is going to make this product druggable and marketable, and what do we need to do to de-risk that product, make sure it's going to fit that marketable perspective, and then also make sure it's going to establish that safety margin. And once we do, then we can sort of say it's worth nominating this candidate and spending real money, right? That's sort of the process. So what is a TPP for a discovery version? You may have heard of a TPP when you get to phase 3. People will build TPPs, they'll actually send them to the FDA. They are sort of very broad, things they have to do with the label claims. A TPP from a discovery version is much more concrete. It's much more definitive. And so the ones that I build, and I built this based on working with several different pharma companies and getting their interplays and kind of building my own. We build it based on things we need to do from an efficacy perspective, things we want to do from a safety perspective, and things we want from a product performance perspective. And each of these have attributes of what we want our product to be. So if we're working in oncology, that's a very different program than if we're working in pain, okay? Oncology, these people are going to die. So we can actually deal with a lot more stringent characteristics. Your therapeutic window doesn't need to be as wide, versus pain is not so much. We don't want to put anyone in, that's a sort of a blockbuster drug. So your target criteria has to really be related to your indication. So we start building these based on what kind of affinity we need to be successful, what kinds of safety activities, whether it's a cardiovascular risk or drug-drug interaction type of risk. And then, of course, in terms of product, does it need to be an oral product? If it's an oral product, you must have bioavailability. And so this helps you identify what your criteria are, and you help work with your med chemists to go through your SAR loops. And so I love my med chemists. I mean, I'm married to a med chemist, but they will just continue to make product and make more compounds forever. They are perfectionists. All they want to do is keep on making more. And so this helps you say, okay, buddy, you're done. You did a great job. We finally got to a compound that is green across the board, or at least mostly green across the board. We're ready to move forward. And so at that point, you are essentially have that point where you've met all your criteria, you've got your therapeutic index, and you're ready to move forward. So that goes back to my nominating the compound. When you've reached the TPP, you're ready to nominate the compound. As far as the therapeutic index, there's a lot of different ways to do this. You can do it based on dose or based on concentrations, but what I've learned is I build these graphs and I continue with them through development. So we start them around late discovery and we keep them around through IND and through early clinical development. As you get more data, certain data trumps, like clinical data always trumps your non-clinical data for the most part. And you keep a track, and this is essentially your PK/PD, sort of understanding of where your TI is from a Cmax perspective or an AUC or a dose perspective, whatever makes the most sense, whatever's most relevant for your particular therapeutic. And you keep track of your TI and make sure you have acceptable window of safety to move your product forward. This is great to have handy as you get ready for partnering discussions, because what happens when you start with partnering and Pfizer comes in the door and they want to look at it, they're going to be going through all your data and they're going to be assembling something like this to basically understand that. So if you have it done yourself, then all the better, because you've established where you think you are. Instead of saying, my drug is safe, now you have proof that your drug is safe. Good? Okay. Now, that was one extreme of I'm safe. The other extreme of hysteria is, oh crap, Michelle, we have tox findings at doses that are too low. We're going to have to kill this program. Where we run a tox study, we have some finding in a dog or a rat or a monkey, and it's not at a dose we expected. We thought we were going to be able to go at 100, and at 10, we saw findings. Is this a deal killer? So the answer is it doesn't have to be a deal killer. And here's why. I'll walk you through it, but here's why. You have found a tox dose that is lower than you expected, but that is just there to help you identify your safe starting dose in humans. And so if you have a way, you can escalate through the equivalent doses past your tox doses in human as long as you have a way to monitor them. So you can actually exceed that. So it does not have to be a deal killer. So let me walk you through that quickly. The first thing is we want to talk about how do you establish starting doses in human trials. The first thing you do is you do your safety program, which is your toxicology studies. You determine your NOAEL, your no adverse effect level. Then you convert it to human equivalent doses based on body surface area. You identify your most sensitive species. Remember, we usually do 2 species. Gene and cell therapies, maybe not so much, but we tend to do 2 species. And you apply a safety factor, and that helps you identify what your first dose is. Sometimes we consider minimally effective doses, and this might be relevant for you because you actually might have a very wide safety margin with your product that's a peptide. And so you can keep going until you hit a limit dose. So in your case, you might actually need to take into effect a minimally effective dose in terms of identifying your safe starting dose. So here's just a quick example. If you don't have this burned into your brain like I do, and you haven't been looking at this guidance, which is estimating the maximum safe starting dose in initial clinical trials for therapeutics in healthy volunteers. It's a go-to guidance. It's very straightforward. It's pretty much set up for small molecules, but it does help you for your large molecules too. You have 2 species. You've identified the NOEL. You've applied this body surface area factor based on this table. And then that gets you a human equivalent dose in the mg per kg unit. In our case, the rat turns out to be the more sensitive species. So that is our conservative choice. So this is our human starting dose. We use the rat dose times the weight of a standard healthy individual. And so we get about 100 mgs. And then you apply your safety factor. And that's how you determine your safe starting dose in humans for your first clinical trial. That's a sub-efficacious dose. Like, we're starting way below where we actually want to get to in our clinical trials. And so I've lined this up on, this is what a standard single ascending dose clinical trial could look like in a normal healthy volunteer. We start at these. Now remember, these are, you finish a cohort, you go to the next. You finish a cohort, you go to the next. These are not all at the same time. These are ascending. You start at that 10 because we identified that as safe. It's well below where we think we're going to see toxicology, but it's sub-efficacious. So we don't want to be there. We need to get into our efficacious region. We start escalating up and up. We're now at 90 mg, which is essentially equal to our tox limit, that ceiling of our tox. We're still sub-efficacious. Remember that? Because we said, oh crap. We're still sub-efficacious. We keep going, and this is okay. We keep going. Now we finally hit what we think is our minimally effective dose. So we're past that tox level. We keep going because we're there. We've now hit what is the lower adverse effect level. This is like your dose limiting toxicity. We're still in the efficacious region, but our people are doing fine. We have toxicology that we can monitor. So we're looking for things and they're still doing great. So we keep on going until we hit that maximum tolerated dose. So that's how you can blast through your tox program. That is generally a fine way to go. There are some divisions where that actually doesn't fly long term. So you might be able to do that, but for different divisions at the FDA, the EU is actually more flexible about this. They will make you stop here at your, essentially your low L or your maximum tolerated dose in your preclinical. So they'll make your preclinical species set a ceiling for your doses. But that's not always the case. So you need to sort of understand where you are, where your indication is, and what division you're working with to figure that out. So the take-home message here is just because you found tox does not mean that you're dead in the water, that there's mechanisms to move forward, especially if you have monitorable tox, which would be like a cardiovascular, like where you can hook them up with a Holter monitor and you can monitor that. So good? Makes sense, everybody? Questions? Okay. FDA won't accept that pharmacology model. I get a question a lot about whether the agency will accept this pharmacology model. And I think that's sort of an interesting question because they don't really care. Typically, it's all about risk versus benefit to them. So as long as you have a data package that supports that the benefit of your product is worth the risk of testing it in humans, then you're generally okay. So the nuances to that is some of the examples I have where, do you guys know what an STZ model is? That is streptozotocin. It's for diabetes. And you actually use this little poison in mice or rats, and you kill the islet cells and kind of damage them. And it's a good model for type 1 diabetes. So we had a program where we were using an STZ data for type 1 diabetes and trying to go into type 2 diabetics. So the agency was like, whoa, you have not actually supported that your drug works in your patient population. And so in this case, we say, well, yes, we did, and we justified why the mechanism of how STZ works kind of worked with our therapeutic, and it was fine. But that's where we got flags on it didn't actually support that. And just another example of xenograft studies for those that do our cancer products. Will a sub-Q flank xenograft study support going into glioblastoma, which is a brain cancer? So if you put something in the flank, will that actually support you doing a brain cancer? And I think the answer is yes or no. It depends on the body of data that you have. If you have 10 different xenograft studies that are all flank, and then several studies that formally show that your drug can get into the brain, then you might be okay. But if you only have one flank xeno study and you go into glioblastoma, I think you're going to get flagged. Because you don't have enough data to support that your drug gets into the brain and that you've reduced the risk for these potential patients. Good? All right. Anybody have questions? One of the myths that we have often is just an assumption here that the tox species for small molecules are rat and dog, And the tox species for biologics are rat and monkey. And that is not the case. You actually have to justify your tox species. And this is tough for those of us who do a lot of budgeting, because if you're like in doing pitch decks and whatnot, you're like, oh, I'm going to do rat and dog, and this is how much that costs. But the truth is, you actually have to determine what the appropriate toxicology species is and then justify it. Once you justify it, make sure it makes sense, then you can sort of budget it. And so you actually need the data to justify it. It's not necessarily a paper exercise all the time. So it's different for different types of products. So small molecules, the standard way to justify the tox species is using comparative metabolism. So comparative metabolism means that you take, usually it's sort of the worst case scenario of liver microsomes, and you apply your therapeutic to that. And in the worst case scenarios, what happens? What metabolic profile do you come up with? And it's very qualitative. It's not quantitative. It's not too expensive. What profile do you come up with? Like, and these are all potential metabolites. These are mass shifts with an M over Z, potential sort of hydroxylation or glucuronidation. These are just potential metabolites. And so what you're looking for is that your combination of your tox species covers you from a metabolic perspective to your human profile. Once you can show that, then you basically have been able to justify for a small molecule that these species, the combination of rat and dog, doesn't have to be perfect, but the combination of the two will actually cover you from a metabolism perspective in your tox program, and that's always been fine. That's not necessarily the case for biologicals. Biologicals don't have metabolic profiles like they do for small molecules. So for biologicals, you generally want to find that your receptor has similar homology between your human and whatever tox species. And that's why people tend to go with monkeys, is because there's a lot of homology between your monkeys. But if you have a highly homologous receptor across different species, then dog should be fine too. So I've had several biologics programs where dog was just as good as monkey, and so if you can avoid it, you do. Just as a note, dogs are less expensive than monkeys, but as you get further in development, monkeys are smaller by a lot than dogs. And so in the short term, it is less expensive to go with dogs. In the long term, it is actually less expensive to go with monkeys, because the drug supply for monkeys is so much lower than the drug supply for dogs. So be careful what you wish for. Antibodies are the same. You want to make sure that your receptor has the same homology, and that the tissue, this is the difference, the tissue cross-reactivity profile for the humans is similar to what you have in your tox species. Gene and cell therapies is a little different. All of these, if you can have a biological response, all the better, but you don't need to necessarily prove it. For gene and cell therapies, having a biological response expected for humans is sort of the way to go for these. And just as a note, you don't necessarily always have tox species the same way you would for a biologic or an antibody. Here you're actually doing your toxicology tend to do your toxicology in your pharmacology model or your transgenic model kind of thing. You're just moving those to super pharmacological doses is what you're doing.